Chapter 9, The Anatomy of the Iris, from Wolff's Anatomy, 8th edition, page number 313. In this video, I'll be talking about the layers of the iris from anterior to posterior, and I'll be talking about the anterior border layer and stroma of the iris. So, there are 4 layers of the iris, and these are anterior border layer, stroma and sphincter muscle. Third is anterior epithelium and dilator muscle, and fourth is posterior pigmented epithelium. This is the microscopic structure of the iris, and it shows the 4 layers. First is anterior border layer or anterior limiting membrane. Second is stroma and sphincter pupillae. Third is anterior epithelium and dilator muscle. Fourth is posterior pigmented epithelium, or it can be divided into 3 layers: anterior border layer or anterior limiting membrane. Second is stroma containing the sphincter pupillae muscle, dilator pupillae muscles, then vessels and nerves. Then epithelial layers, that is the third layer, containing anterior pigmented epithelium and posterior heavily pigmented epithelium. Both the layers of the epithelium are pigmented, but posterior pigmented epithelium is heavily pigmented. Anterior border layer. The anterior border layer is a condensation of connective tissue and pigment cells derived from the anterior stroma. Fibroblasts form a fairly continuous sheet of flat stellate cells and interlacing processes stretching from the iris root, where there is continuity with the ciliary body stroma, to the pupil, where they reach the epithelium of the pupillary roof. The fibroblasts exhibit microvilli and cilia which project into the anterior chamber. Pigmented uveal melanocytes, also oriented parallel to the surface, lie deep to the fibroblasts. In about 57% of the eyes, the anterior border layer extends into the drainage angle as fine iris processes, which insert anterior to the scleral spur and sometimes up to Schallbase's line. 3 types of intercellular junction are reported to join the cells of the anterior border layer, including gap junctions, intermediate junctions, and discontinuous tight junctions. There are how many types of the junctions? Zonular occludence, tight junctions, then adherents, zonular or macular adherents, then desmosomes, and hemidesmosomes and gap junctions. So in between cells, there are 4 types of the junctions: tight junctions, that is occludence, then adherents, then comes desmosomes, and then gap junctions. Okay, gap junctions are usually leaky and they can be termed as zonular or macular. If it is a dot like point, it is connecting the 2 cells, then it is called as macula, and it is zonular when it is zonular strap that's connecting the 2 cells. It is called as zonular. Then zonular occludentis, macula occludentis, then zonular adherents, macula adherents, like that. Desmosomes and hemidesmosomes, they connect the cell to the basement membrane or extracellular connective tissue. Okay, so I think zonular adherents is nothing but, I think macular adherents is nothing but the desmosomes. Yeah, I have to confirm that thing. As far as I remember, last time I had read it, it is the macular adherents that is equivalent to desmosomes. So 3 types of intercellular junctions are reported to join the cells of the anterior border layer, including gap junctions, intermediate junctions, and discontinuous tight junctions, where between the fibroblasts and the melanocytes present in them. So all of these junctions occur principally between the cells of like types like fibroblast, and melanocytes and melanocytes. With the exception that gap junctions have been shown to exist between fibroblasts and melanocytes in one species of monkey. In monkey, not in human beings, there are gap junctions between the melanocytes and fibroblasts of the anterior border layer. So capillaries and venules are present in this layer, as well as numerous nerve endings which terminate in the subjacent stroma. Nevus-like clusters are probably derived from Schwann cells. Small elevated white or yellowish Wolffian spots, not Wolffian, Wolffian spots, W-O-L-F-F-L-I-N, Wolffian spots are found in the anterior border layer at the iris periphery. So So Wolfflin spots, Wolfflin. Wolfflin spots are what? These are small, elevated, white and yellowish, found in the anterior border layer at the iris periphery, most often in blue and gray irides. So Wolfflin spots. These are present at the periphery of the iris, anterior border layer, but in the blue and gray irides. The border layer is absent at crypts and thinned at contraction fluids. It is thickest in the pupillary zone and at the periphery of the ciliary zone. The layer is responsible for the color of the iris, is thin in the blue iris and thick and densely pigmented in the brown iris. And densely pigmented, once again. The layer is responsible for the color of the iris, is thin in the blue iris and thick and densely pigmented in the brown iris. Melanocytes may accumulate locally to form an iris freckle or may be distributed more diffusely to produce a patch of hyperheterochromia or heterochromia of one iris in comparison to the other. So what we have learned in anterior border layer, it's made up of what? It is the condensation of the connective tissue, condensation of the anterior part of the stromal connective tissue first thing. Then it has got cells, so it has got connective tissue and cells, which cells fibroblasts and the melanocytes. Now fibroblasts, beneath the fibroblast layer, there is a layer of the melanocytes. So fibroblasts, how they are arranged, they are stellate shaped. They have cytoplasmic processes, they form the flat sheet, they are connected to each other with the cell junctions like these gap junctions, intermediate junctions, and discontinuous tight junctions, means adjuvants, occludents, and gap junctions, and desmosomes. But there are no junctions in between the fibrocytes or fibroblasts and the melanocytes. That is the second point. Then third point is Wolfflin spots. Wolfflin spots are the yellowish, white, elevated small spots which are present in the anterior border layer at the periphery of the iris in the blue and gray irides. And this anterior border layer, it is thin at the contraction furrows and it is the thickest at pupillary zone and the periphery of the ciliary zone, and it is absent over the crypts. Once again, the anterior basement membrane or anterior border layer of the iris, it is absent on the crypts. It is thinnest or thin over the contraction furrows, then it is thickest in the pupillary zone and at the periphery of the ciliary zone. And this layer is responsible for, because it has melanocytes, melanosomes, melanin granules, so this layer is responsible for the color of the iris along with the stroma, because stroma also has melanocytes, we will see it later. Then this layer, that is the anterior border layer, it is thin in blue iris and thick and densely pigmented in the brown iris. Now melanocyte aggregation can form the iris freckle in this layer, or their distribution, even distribution in between the two, isomer distribution, causes the similar color of both the irises. Otherwise, it leads to heterochromia or hyperheterochromia depending upon the pigmentation. So it's because of these melanocytes in this layer also in the stroma. That's the other thing. Now again, stroma. Second point, first layer is done, anterior border layer, anterior limiting membrane. Second layer is stroma. We will cover the stroma, like 20% part of stroma will cover today because it's a vast topic. So tomorrow I will complete stroma, the second part of the stroma. That is the fourth part of the iris anatomy. Now stroma. The stroma consists of loose collagenous network containing the sphincter pupillae muscle, the vessels and nerves of the iris, cellular elements, fibroblasts, melanocytes, clump cells, and mast cells. So cellular elements, fibroblasts, melanocytes like anterior border layer, additionally clump cells and mast cells, then vessels and nerves and connective tissue. The collagen bundles are interlaced and are arranged as curved clockwise and anti-clockwise arcades. This arrangement is less regular than in birds and lower primates. In birds, the lower primates and other mammals. The bundles are condensed around vessels and nerves and have an annular orientation near the pupil. The fibers are attached to the iridium muscle, the anterior border layer and the ciliary stroma. So these collagen fibers present in the stroma, these are arranged in annular fashion. And second thing, these are attached to the iridium muscles, that is sphincter pupillae muscle, dilator pupillae muscle. Then ciliary stroma and anterior border layer, anteriorly to the anterior limiting membrane. Then these collagen fibers are attached anteriorly to the anterior limiting membrane. Then in the stroma, attached to the muscles, sphincter pupillae, dilator pupillae muscles, and also to the ciliary stroma. There is no elastin in this stroma of iris. There is no elastin. It's a collagen tissue, collagen fibers. Then white spaces exist in the stroma containing a hyaluronidase sensitive glycosaminoglycan. There is a glycosaminoglycan that is sensitive to hyaluronidase. This loose arrangement permits a free diffusion of aqueous and large molecules into the stroma and probably facilitates the pupil movement. So there are white spaces in the stroma and they contain the glycosaminoglycans, that is glycans, that is hyaluronidase sensitive. And there is a loose arrangement of the iristomal collagen fibrils and it allows the free diffusion of the aqueous and the large molecules into the stroma and probably facilitates pupillary movement. So now we'll see the figures. Figure 9.16, cross-sectional view of iris and ciliary body. The anterior border layer AB forms a distinct zone at the iris surface. The iris dilator muscle ID, derived from the processes of the anterior myoepithelium, lies deep to the sphincter muscle. As it approaches the pupil, IR stands for iris root. Note the continuity between the root and ciliary body structure. IS for iris sphincter, IB for iris vessels, PE posterior pigment epithelium, PR for pupillary ruff, curling around the pupil margin, CP for ciliary processes. So you see the anterior border layer here, that is shown as AB, that is a distinct layer. Then there is a stroma. In the stroma you will see IB means iris vessels, then IS means iris sphincter, sphincter pupillae muscle, and deep to the sphincter pupillae muscle you will see the area, that is iris dilator pupillae muscle. Then comes the IR. IR is what? Iris root, and after that you can see the ciliary processes there, posterior to IR. Then you will see the pigment epithelium, PE, that is shown as dark brown in color here. So that was about figure 9.6. Then figure 9.17. Anterior layers of the iris. The anterior border layer is covered by a single layer of fibroblasts A, whose long branching processes interconnect with each other. The branching processes of the fibroblasts form very sized openings on the iris surface. Beneath the layer of fibroblast is a fairly dense aggregation of melanocytes and a few fibroblasts again. The superficial layer of fibroblasts has been removed at B to show the cells. The number of cells in the anterior border layer is greater than in the underlying stroma. The iris stroma contains a number of capillaries, which sometimes are quite close to the surface. So this is the anterior border layer, which is made up of the fibroblast, a flattened sheet of the fibroblast interconnected to each other. These are stellate shaped, they have cytoplasmic processes and connected to each other by the junctions, tight junctions, intermediate junctions and gap junctions. Then beneath the fibroblast layer, they have removed the fibroblast layer cover for our convenience to see the melanocyte layer beneath, that is shown in the figure B, and B is very faint here. that is below the melanocyte word. It's there, B. So you can see the melanocytes, and along with the melanocytes, few fibroblasts are present there in this layer beneath the fibroblastic layer. Okay, so that was about the anterior layer, fibroblasts and melanocytes. Then figure 9.18, anterior border layer electron micrograph. The thin layer of fibroblasts on the anterior artery surface A is not continuous. Melanocytes B and their processes C form part of the anterior border layer. The Golgi apparatus D and melanin granules E are shown. So you can see here the fibroblastic layer. It is not continuous. Obviously, it is not continuous. They are interconnected, but this is not a continuous layer. And melanocytes here showing the melanin granules and Golgi apparatus also. So that was about the figure 9.18. The figure 9.19. The anterior border of the iris is composed of discontinuous layer of fibroblasts and interspersed collagen fibrils. Original magnification 13,800 times. Insert shows fibroblasts and a meshwork of collagen connection fibroblasts. Okay, so the anterior border of the iris is composed of fibroblastic layer, then beneath that melanocytes and few fibroblasts again. Then it is made up of the collagen and meshwork of the collagen that is connecting the fibroblasts. Okay, so fibroblastic cells are interconnected to each other, but these are not continuous. In between them there are spaces and these are filled with the collagen tissue. So figure 9.20. Insert shows a junction complex arrow forming the anterior border layer of the iris. Scanning electron microscopy, original magnification 1500 times. Melanocytes M are present beneath the fibroblast F of the anterior border layer. Original magnification is 10,500 times. So once again, figure 9.20, insert shows a junction complex forming the anterior border layer of the iris. Scanning electron microscopy, original magnification is 1500 times. Melanocytes M are present beneath the fibroblast of the anterior border layer. Okay, so you can see the fibroblast F here, melanocytes beneath the fibroblast M, and you can see the junctions between the fibroblasts here. Yeah, junction complex shown by arrow between the fibroblastic processes. Now figure 9.21 showing the section of pupillary margin. Pupillary margin showing the pupillary rough. You can see the pigmented epithelium that is just curving, trying to curve anteriorly, but it doesn't lie on the anterior surface of the iris. When it does that, it is called as iris ectropion, but it is not the iris ectropion here. It is just projecting forward so that it is visible at the pupillary edge, not on the anterior surface of the iris. That is the pupillary lobe. That is the extension of the pigmented posterior pigmented epithelium of the iris at the pupillary edge. So that it is visible. That is pupillary lobe, and it is created because of the radial folds on the posterior surface of it. Now, figure 9.22, meridional section of posterior layer of the iris. So you can see meridional section of the posterior layer of the iris. You can actually see the layers of the iris here. You can see the clump cells, then sphincter of sphincter pupillae muscle, periphery of the sphincter pupillae muscle, dilated pupillae muscle, and posterior pigmented cell. Sphincter pupillae, capillary plexus. So it is showing the iris stroma and the posterior pigmented epithelium. The stroma consists of the clump cells, mast cells, melanocytes and fibroblasts, and the collagen fibrils from the elastic fiber. These collagen fibrils, they have white spaces in between, and there is extracellular matrix that is hyaluronidase positive, glycosaminoglycans in it. Then after that, these collagen fibrils, they have white spaces in between to allow the free flow or diffusion of water and some molecules, relatively large molecules, to facilitate the movement of the iris. Also, these collagen fibrils are attached to the anterior border layer, then muscles of the iris, it is dilated and sphincter muscles, and also to the ciliary body stoma. And these this connective tissue of the iris is devoid of the elastic. So that was about the stoma of the iris in short. Tomorrow we will cover the sphincter muscles, then dilator pupillary muscle, then vessels of the stoma of the iris. Okay. So this will be covered in either one part or two parts. Enough for today. Thank you so much.